Great. So welcome, everybody. I'm Cudkowicz. We're here today to hear about the science of our new regimen, Regimen F by Calico and ABBV, and to have a chance to hear your questions and try to answer them as well. Today, what we're going to start with a science presentation by Calico. It's pre-recorded, and we'll play that. And that's about 15-20 minutes, and then we'll have the rest of the time for Q&A. Please put your questions in as you hear the recording or anytime that you want, and we'll try to get to all of them. We have a lot of people on the panel today, and after the video, as people answer questions, they'll present themselves. But we have people from Calico as well as Dr. Driss, who's the co-lead of this particular regimen, Dr. Senda Ajroud-Driss from Northwestern, and other people also from the HEALEY Center. We will record everything and then post it on our website so that if you want to rehear anything, or if other people haven't been able to make it today, they can listen to it as well. And with that, I think we'll go straight to the recording.
Hello, everybody. My name is Bill Cho, and I am the head of clinical science at Calico, and I'd like to introduce to you ABBV-CLS-7262, which is a new investigational agent that's currently in development as a potential treatment for ALS. And I'm very excited that 7262 has been selected as a Regimen F to participate in the HEALEY ALS Platform Trial, and Regimen F is currently enrolling. So first, I'd like to say a few words about Calico, since we're a relative newcomer to the biopharmaceutical world. Calico was founded in 2013 by Google, now known as Alphabet, and Art Levinson, who is the former CEO of Genentech, and we have an ambitious mission, which is to understand human aging and to develop therapies for age-related disorders, including neurodegeneration. We're in a partnership with ABBV, which is a global biopharmaceutical company with a proven track record of developing medicine and solutions for people living with neuropsychiatric disorders such as Parkinson's disease, schizophrenia, and depression. So in our pursuit of trying to understand the biology of aging, we find ourselves investigating some fundamental biological pathways. And one such pathway is a pathway called the integrated stress response, or the ISR. So the integrated stress response, or the ISR, is a very important biological pathway, and we know it's important because it's used throughout nature. And it's found in almost all cells and most living organisms, and it's a pathway that allows cells to handle various types of stressful situations such as nutrient deprivation. The two key players in this pathway are EIF2 and EIF2B, which are enzymes that in normal situations work together to help generate the proteins that are required for the normal function of cells. However, when a cell encounters a stressful situation, EIF2 becomes modified, and that's depicted by the addition of this P, or a phosphate group, on EIF2. And when this occurs, EIF2 and EIF2B no longer work together to make proteins, but EIF2, that's phosphorylated, actually reduces the activity of EIF2B. And as a result, normal protein synthesis is reduced, and instead there's an increase in stress proteins, which can be harmful to the cells. In addition, the stress causes the formation of stress granules that contain a protein called TDP-43. And in most situations, the stressful situation is temporary and the cell can go back to its normal state. But when a cell encounters persistent stress or severe stress, this pathway, the ISR, is chronically activated, which results in the chronic formation of stress proteins. And also we believe that the TDP-43 found in these stress granules can further accumulate into these aggregates of TDP-43 that don't disappear. And many of you have probably heard about this protein, TDP-43, because it is a protein that's strongly associated with the causes of ALS. So let me tell you a little bit more about why we believe the ISR is involved in ALS. We have observed that the ISR is activated in ALS, and this observation came from examining brain and spinal cord samples from people who died of ALS. And to remind you, we believe that ISR activation in these brain and spinal cord cells will lead to a decrease in normal protein synthesis and an increase in stress proteins and stress granules that contain TDP-43 that can be harmful to the cells. And we believe that the TDP-43 found in these stress granules can eventually lead to the further aggregation of TDP-43. These TDP-43 aggregates have been found in the vast majority of instances of ALS. So I've told you that chronic activation of the ISR is detrimental to cells, and we found evidence that the ISR is activated in ALS. So can the ISR be inhibited? The first major breakthrough came from the discovery of a molecule called ISRIB, or ISRIB, which is short for ISR inhibitor. And this molecule was discovered by a scientist named Carmela Sidrowski while she was working at the University of California, San Francisco. Carmela has continued her work on the ISR at Calico, and she's actually here with us today. And the way that it was discovered that this molecule inhibits the ISR is that it actually binds to EIF2B. It turns out that EIF2B is a very complex enzyme made of multiple smaller proteins, and ISRIB binds in the middle of this complicated enzyme and increases the activity of this enzyme. And it also changes the shape of the enzyme, making it less sensitive to the effects of stress. So let me quickly recap how EIF2B is involved in the ISR. So recall that in stressful situations, EIF2 becomes phosphorylated, and this phosphorylated form of EIF2 reduces the activity of its partner EIF2B. And as a result, there's a decrease in normal protein synthesis, and there's an increase in the synthesis of stress proteins and stress granules that are harmful to the cells. And what ISRIB does is that it boosts the activity of EIF2B, and it also makes EIF2B less sensitive to the effects of stress. And the net result is that there's a restoration of normal protein synthesis, and there's a reduction in the formation of harmful stress proteins. So these are some of the early experiments that were done to prove that ISRIB is having the effect that we hypothesize. And in these experiments, we're measuring the amount of the stress protein called ATF4. So this experiment was conducted in 3 parts, and if you focus your attention to the leftmost column in this experiment, these are cells that are in their normal resting state. They're not exposed to stress, and there's no drug on board. And in this condition, there is no ATF4, the stress protein ATF4 that's made. So when these cells are exposed to stress, you can see that the stress protein ATF4 is significantly increased. And when these cells are treated with ISRIB, the production of that stress protein ATF4 is significantly reduced. So since the initial discovery of ISRIB, the Calico and ABBV scientists have been working hard to develop next generation molecules, which we call EIF2B activators, and we've tested these molecules in a variety of biological systems. So let me first describe to you some of the experiments that were done with these 2B activating compounds in vitro. And what I mean by that is that these are experiments done in isolated cells outside the body. And the particular cell type that we studied in these experiments are neurons, which are the cell types that reside in the brain and spinal cord. And this is a very specific type of neuron called a motor neuron cell. And this is the type of cell that controls muscle activity and also the type of cell that's damaged in ALS. So we can take these cells and expose them to stress, and when this happens, you see the formation of the stress granules that I described to you earlier, and you can clearly see the stress granules in the red circle in the microscope image on the left. And when these cells are treated with 2B activating compounds, these stress granules disappear within minutes and also can prevent the formation of new stress granules. So we next wanted to conduct experiments to see whether or not these 2B activating molecules would have an impact on a living animal, not just cells in a dish. And so we decided to study mouse, but there are no good mouse models of ALS, and so we created our own model. And in this model, we've mutated EIF2B. And this mutation, we know from previous experiments, activates the ISR. And it also causes problems in the structure of the nervous system, in the spinal cord and in the brain. And it also produces problems with the mouse's behavior. They are incoordinated and they have general problems with movement. So we thought that this would be a great model to see whether or not these 2B activator molecules could have an impact and fix some of the problems in this mouse that has a defective EIF2B. So let me first walk you through some of the effects that we saw on the structure of the spinal cord. So these are microscope slides that were generated looking at the spinal cord in these mice. And the leftmost image shows what a normal mouse spinal cord looks like. And these are stained to highlight a particular cell type in the spinal cord called white matter cells. And on the left, you can see the normal organization of the spinal cord. The mutated spinal cord in the middle panel shows that there's a significant reduction in this particular cell type, and the cells are organized differently. But when these mice are treated with 2B activating molecules, you can see that there's a dramatic increase in the number of these cells that are defective in the mutated mouse, and also the organization of these cells looks much more similar to what you saw in the normal mouse. So we see that these 2B activator molecules can fix some of the structural problems that are seen in the nervous system of these mice. But we also wanted to see whether or not they could fix some of the behavioral problems. And so I previously told you that this mouse has problems with coordination and walking. So we wanted to see whether or not our 2B activating molecules would have an impact on this type of behavior. So in this experiment, we ask mice to cross a balance beam, and we can measure the amount of time it takes to cross the balance beam. So on the y-axis of this graph, this is the time to cross the beam, and on the x-axis we have the age of the mouse in the experiment, and the ages of this experiment range from 24 to 35 weeks. So the bottom line, the black line, shows the results of looking at a normal mouse, and you can see that the normal mouse can cross that balance beam in a matter of a few seconds, maybe around 5 seconds, and it's a steady pace all the way throughout the course of the experiment. Now if you compare that to the behavior of the mutant mouse, shown in the gray line, you can see that starting around 24 weeks of age, it takes the mice a little bit longer to cross the balance beam, which reflects some of the problems with its movement and coordination. And as the experiment goes on, as the mice get older, it takes even longer and longer to cross this balance beam. So that behavioral problem gets worse. When these mice are treated with 2B activator compounds, you can see that initial, and this is the line shown in green, you can see that the initial problems with the walking speed that show up at 24 weeks get better with time on treatment with this molecule. And eventually, towards the end of the experiment, they're able to cross that balance beam in nearly the same amount of time that it takes for a normal mouse to cross. So we next wanted to do a set of experiments to see whether or not these 2B activating molecules would have an impact on conditions of extreme stress, stress that's so severe that the cells die very rapidly. And so we conducted a set of experiments, again in mouse, in a mouse model called the optic nerve crush injury model. And others, scientists, have demonstrated that this injury is caused by activation of the ISR. So in this experiment, the nerve fibers that come out of the eyeball are crushed with a set of forceps, and this crush injury causes the nerve fibers to die back, and eventually the nerve cells in the retina that gives rise to these fibers, they die very rapidly. So we wanted to see whether or not the 2B activator molecules would have an impact on this injury model. So these are some of the experiments, experimental results shown in the graph on the right. And what's plotted on the y-axis is the number of surviving retinal ganglion cells. And the white bar on the very left portion of the graph is the normal number of retinal ganglion cells without any injury. And then the gray bar to the right of that shows that there's a significant reduction in the number of living retinal ganglion cells after the injury. And the two green bars show what happens when these mice are treated with 2B activator molecules either before the injury, shown in the dark green bar, and after the injury, up to 6 hours after the injury. And in both conditions, we can rescue many of these neurons from dying as a result of the nerve injury. So the results that I just described to you provide evidence that our eIF2B activating molecules are indeed active in various stressful conditions impacting cells in a dish up to a living mouse. But before moving on, I just want to quickly recap our mechanism. So by now you should be somewhat familiar with this schematic. In conditions of chronic stress, which we believe is occurring in ALS, the ISR is persistently activated, and when the ISR is persistently activated, there's a decrease in the production of normal proteins and an increase in the production of stress proteins that can be harmful to the cells. And in addition, when the ISR is chronically activated, this can lead to the formation of TDP-43 aggregates that are known to be associated with most instances of ALS. And so what 2B activator molecules like ABBV-CLS-7262 do is it disrupts the ISR by boosting the activity of eIF2B and making eIF2B less sensitive to the effects of stress. And this causes a restoration of normal protein production, a reduction in the amount of stress proteins that are harmful to the cells, and dissolving stress granules that may lead to TDP-43 aggregates. So as we were preparing for clinical trials, we picked the molecule that had the best drug-like properties, and as you know, this molecule is called ABBV-CLS-7262. So we began clinical studies in healthy volunteers in what are known as phase 1 studies. And the purpose of phase 1 studies is to understand the safety and tolerability of new molecules going into people for the first time and to understand how these new molecules behave inside the body. So in these phase 1 studies, we administered ABBV-CLS-7262, or 7262 for short, to over 100 healthy volunteers across a wide range of doses. And what we learned is that 7262 can be given by mouth once a day. The side effects or adverse events were non-serious, and all of the side effects were mild to moderate in severity. We were able to demonstrate that 7262 increased eIF2B activity and inhibited the ISR as expected in the blood cells collected from our healthy volunteers. And then finally, we were able to measure the amount of drug in cerebral spinal fluid, or CSF. And this is the fluid that surrounds the brain and spinal cord. And we were able to identify the doses that contain sufficient concentrations of 7262 in this fluid that we believe will be active in suppressing the ISR. So in the last slide, I mentioned that we were able to demonstrate that 7262 is active in the blood cells collected from our phase 1 healthy volunteer trial participants, and this is a summary of those results. So first, let me remind you that eIF2 and eIF2B are enzymes that work together to make proteins, and this protein production requires energy. And the energy comes from a molecule called GTP, and the energy is chemical energy stored in GTP. And what eIF2 does is it breaks down GTP to GDP, releasing that energy that's used towards protein production. And what eIF2B does is it exchanges GDP with a fresh molecule of GTP. And this continuous exchange of GDP to GTP provides a continuous energy source for protein production. And we can measure the reaction rate, the exchange rate of GDP to GTP, and that's what's shown in the experiments shown here. So on the y-axis, we're plotting the reaction rate, and on the x-axis, we're showing the results from blood cells taken either 6 hours or 24 hours after dosing. So at 6 hours after dosing, we show that 7262 is able to increase that reaction rate, depicted in the green bars, relative to the blood cells that were collected from people given the inactive compound or placebo in the gray bars. And we think that this is a fairly long-lasting effect because this eIF2B stimulation is also seen in blood cells collected 24 hours after dosing. So we're able to demonstrate that 7262 can stimulate eIF2B activity for a long time after dosing. So we also wanted to demonstrate that 7262 can inhibit the ISR. So in this set of experiments, we measured the ISR product called TRIB3, and we plot the amount of TRIB3 on the y-axis. And again, we look at the conditions that in blood cells that were collected either 6 and 24 hours after dosing. And in the blood cells from people given 7262 in the green bars, we see a reduction in this ISR product, TRIB3. And this, again, seems to be a long-lasting effect because it's also observed 24 hours after dosing. So these results demonstrate that we were able to inhibit the ISR in the blood cells. After completing these phase 1 trials in healthy volunteers, we moved on to initiating clinical trials in people living with ALS. Now, these are some of the safety results that are coming out of that ongoing trial in people living with ALS. We intended to enroll 30 people, but we ended up enrolling 31. And some of the first people that entered the trial have been on the study for almost a year now. And the most common side effects that we've observed so far include headache, nausea, itchiness, constipation, and dizziness. And because this trial is ongoing, we can't yet say for certain whether or not these side effects were caused by 7262 or the inactive placebo compound. So given the safety profile to date, we're ready to take the next step, and we are very excited that ABBV-CLS-7262 will be the next regimen, Regimen F, in the HEALEY ALS Platform Trial. And the way that Regimen F is designed is very similar to previous regimens in that there will be first a 6-month placebo-controlled randomized period where trial participants will receive either active drug or placebo in a 3-to-1 fashion. And this means that participants are 3 times more likely to receive active drug than placebo. One important difference here is that the active drug, for those participants who are assigned active drug, they will be divided into 2 different dose levels. Both of these dose levels are hypothesized to activate EIF2B. Following the 6-month randomized period, participants then can roll over into an active treatment extension and participate in this active treatment extension for approximately 1 year. So one important aspect of Regimen F is that we will be collecting biomarkers throughout the course of this study. And biomarkers are substances in the body, and these substances can be proteins or chemicals or genetic material that can really help us better understand the causes of ALS, and more specifically may help us understand the role of the ISR in ALS. One great example of what we may learn is that there are people with different levels of ISR activation at the beginning of the study, and they may predict which people are more likely to respond to the effects of 7262, and this information will be very important for future clinical trials. So we'll be collecting blood periodically throughout the course of the study, and then we'll also be collecting CSF at the beginning and at the end of the 6-month randomized control period. And now I just want to summarize. So Calico and ABBV are very excited that ABBV-CLS-7262 is now ready to be evaluated as a new potential treatment for ALS by inhibiting the ISR. And we're very enthusiastic about this mechanism because we have observed that the ISR is activated in ALS, and we know that 7262 is a potent inhibitor of the ISR by binding to and activating the enzyme EIF2B. We know that aggregates of the protein TDP-43 are observed in most instances of ALS, and we demonstrated that 7262 can quickly dissolve stress granules containing this protein TDP-43. New drugs tested in ALS clinical trials should have their intended biological effect in people, and we've demonstrated this by collecting blood cells from our phase 1 trial participants and showed that 7262 can increase EIF2B activity and reduce the ISR. The right dose needs to be administered in clinical trials, and we've measured 7262 in the CSF at levels that we believe will activate EIF2B. Despite decades of research, the understanding of ALS is still incomplete, and we hope to contribute to this understanding by collecting biomarkers in CSF and blood that will help us improve our understanding of ALS, help us understand how the ISR is involved in ALS, and may identify people who are more likely to respond to 7262. Thank you for your attention, and now several members of the Calico team are here to help answer any of your questions.
Thank you, Bill, for your great explanation. I do see 2 questions already in the Q&A. Should I read them? Okay. So the first question is, did you test the 7262 on both the upper and the lower motor neuron? And Bill, if you can answer
So
that.
yeah, maybe I can start trying to answer that question. So first of all, that's a fantastic question. It's a great question. I just wanted to clarify that in that particular slide where we showed the photomicrographs of neurons, those were human iPSC derived motor neurons. So those are neurons that are grown from a pluripotent cell. It's a cell type that can differentiate into multiple types of cells, and we can induce these cells to turn into motor neurons. And so there is no real distinguishing differentiation between upper and motor neurons in that system.
Very good. There is a question that you probably answered at the end of the video is, has this drug been given to healthy subject or ALS patient? So to this date, this drug was given to 100 healthy volunteer and 31 ALS patient in a phase 1 study. And since the phase 1 is still ongoing, we do not have result to share. And I'm answering the other question that came in after that. There is a very good question here asking, is there any anticipated difference for patient with sporadic versus genetic ALS?
Yeah, so maybe I can start answering that question. So we don't have specific data to tell us whether or not there's going to be a greater effect in sporadic versus familial forms of ALS. We know that TDP-43 aggregates are found in the majority of cases of ALS. So we anticipate that this would be potentially helpful for a large number of people with ALS. And, you know, the ISR mechanism, we believe, is sort of downstream of many of the initiating causes of ALS, regardless of the etiology. So we're hopeful that this will be potentially beneficial in a wide variety of people with ALS. Is there anybody else that might want to add to that? Open it up.
I'll just add that for this platform trial, this is open to sporadic and familial, and we do think that this drug would work in all forms of ALS. The only two forms of ALS that don't have TDP-43 aggregates, and this is still a item of debate in the field, is maybe SOD1, people with SOD1 ALS or with FUS ALS, but that doesn't mean that integrated stress response isn't involved in those two. But everybody else with ALS has the same TDP-43 biology. So we really do think that this is a regimen for all people with ALS.
Another question is there an EAP for this regimen? And if no, why not? Maybe I'll direct this to the Calico team, or?
Yeah, thank you. And I think Emmanuel Bellarmine, who's our head of regulatory affairs and quality assurance, can maybe help answer that question.
Yeah, thank you, Bill. And that's another excellent question. So since 7262 is a new investigational drug with a new mechanism of action and is not yet approved in any countries, its use is currently restricted to clinical trials where we can monitor the safety of each participant. And at this point, even though 7262 appears to be adequately tolerated in healthy volunteer and people with ALS, we are still collecting incremental safety information. But having said that, we are considering a limited EAP no sooner than after the first futility analysis in the HEALEY platform, and once the results of the placebo-controlled portion of the HEALEY platform are going to be available for Regimen F. Depending on these results, we may consider a larger EAP for our drug.
Good, thank you.
Yeah, and in accordance with the applicable regulation, I mean, the EAP will be intended for people with ALS who are not eligible to participate in a clinical trial with 7262.
Yeah, I might expand on that this approach is exactly the approach taken by all the other regimen partners, is really waiting until we have some of those interim looks at the data for futility, which is really just making sure that it's safe before opening up a limited EAP for people not eligible for the study.
Very good, thank you. There's another question about ALS meds and if we can still take them during this study. And I can answer this. Regimen F, to qualify for Regimen F, we need to qualify first for the master protocol. So the same inclusion criteria will be used like prior regimen, and all FDA-approved drugs for ALS will be allowed. The same prohibited medication will remain, and we usually have a list of those, and we'll share them with the patient. So some of the medication that are still in trials, for example, or investigated for ALS will not be approved to be used during the regimen F, for example. And this is the same thing we did for the other regimens. There's another question. If I participate in regimen E, can I have access to this drug? Merit, if you
I'm happy to answer that. So if somebody's in regimen E and they've finished, the double blind, or they've decided not to go into open label, or they finished that, they're still eligible for the platform trial, they could come back in. So there would be a wash-up period, which is about 30 days of being off of regimen E and then going back to the master level for screening if you're still eligible. And thank you again for being part of E and thinking about being part of another regimen.
Very good. This question may be for the Calico team. What markers will be measured in the blood and the CSF? And how often will be the blood drawn, the blood work be done?
And then maybe I can hand that over to Amos, who's our head of biomarkers at Calico.
Hey, hi everyone. This excellent question. So there's several classes of biomarkers that we're going to measure. Those are extremely important to really understand how the drug works. So in CSF, we'll focus on measuring biomarkers of the ISR to tell us whether the ISR is on and whether it's off with the drug. So then again, would tell us whether the drug is doing what it's supposed to do. Measuring the ISR at baseline would also tell us which patient might benefit mostly from this therapy. Obviously, ALS is a heterogeneous disease, and it could be that the level of ISR in different people with ALS may result in different response to the drug. And then finally, we're going to measure biomarkers that are indicative of neuroinflammation as well as neurodegeneration. So those would tell us how effective is the drug in patients, and then would actually used as part of the totality of evidence to tell us whether things are working.
Very good. It is a very good question asking about external stress and cellular stress. We don't know if external stress may be contributing to cellular stress, but we are focusing with the study we are focusing on cellular stress. That may be a little bit different. Outside stress is always bad anyway, so any way to mitigate that would be good. Is there any expected drug interaction with this therapy? And will we be able to administer it in feeding tubes? I can answer that too. There is a list of medication that will interact with this compound. They're not usually used by ALS patient, but we'll make sure that when we talk to the patient about the study, we go over that and make sure they're not taking any medication that will show any interaction. And if the patient is no longer able to swallow, the compound could be administered through the feeding tube. Let's see. There's a question here. You showed safety result from phase 1 in ALS patient. Did you also measure biomarker in ALS patient? Like, do we have already some information about the biomarker that were measured during the phase 1?
Yeah, so maybe I can take that as well. So we have looked at the biomarkers that were proposed to measure in the HEALEY platform trial. Those obviously were characterized very well, both in preclinical models as well as in samples from people with ALS, both CSF and plasma. And we as to corroborate some of the things that Bill showed you in terms of ISR elevation, we do see elevation of specific ISR related biomarkers in cerebrospinal fluid from people with ALS.
There's a question here asking if the HEALEY trial master protocol in clinicaltrial.gov will be updated with the latest site-specific information regarding contact and recruitment status.
Yes, absolutely. We're going to update with all the sites that are activated as they're getting activated, and also update some of the changes we've had in the master protocol and the regimen specific inclusion exclusion criteria. We currently have, I think, 18 sites activated, which is fantastic. And of course, we also on our Thursday webinar at 5 to 5:30 every week update everybody there as well.
Thank you, Merit. And there is a question here about what is the time cutoffs in the start of ALS. And again, as I said, I mean, the inclusion criteria are the inclusion for the master protocol from the HEALEY platform trial. So 3 years from the onset of the disease, weakness, or speech issue. And I see two questions asking about reversal. Is it anticipated that this drug may reverse some of ALS symptom rather than just slow progression?
Either, I think we don't know. I mean, we have designed the study, empowered it, meaning having enough people in the study to be able to pick up a slowing of the rate of progression. But obviously we're going to be looking to see how big effect that is in this study. I don't think anyone, to be honest, would be able to tell anybody living with ALS today, other than perhaps people with the genetic form of the illness, that we have a treatment that is going to stop or reverse your illness. I mean, that is all our goal, but we need to really understand the biology better. I do think that the biology behind this drug is fantastic. It's really targeting something that we know is relevant for all forms of the illness. But we don't know. This trial will tell us, the effect in people with ALS. Is really a very important trial.
Yeah, there was two questions about technical difficulties. Apparently the sound kind of is choppy just for the technical team.
I'm sorry about that. We'll just post this, and I think most of it, I could hear everything. It's just a little difference in volume, but we'll post it and see if we can improve it before we post it.
And there is a question about if all the sites are open to enrollment. We're getting there. We're actually activating sites as quickly as we can, and we're hoping that all the sites will be open pretty soon. This question is for the Calico team. They're asking, what is the reason for 2 doses? Is it for efficacy or adverse event?
Oh, in the platform trial, yeah, we are planning to explore 2 different dose levels. There is one dose that, and I just want to clarify that both of these dose levels are expected to be fully engaging the target and should have the intended effect on EIF2B. We do have a standard dose, and that's the bulk of the people who will be on active drug will go into that standard dose. But we're also exploring a higher dose level just because the safety profile so far that we've encountered supports testing a slightly higher dose level to see whether or not there might be any additional benefit that comes from exploring that higher dose level, either in clinical efficacy markers or in the biomarkers. So we do want to sort of expand the range of doses to be explored in this trial. It's a great question. Thanks.
Thank you, Bill. How many will be in the next trial, and will the CSF draw be a requirement? So usually we have enrolled 160 patients in every platform trial so far, but for this next one, we're going to enroll 240 patients, a little bit higher number, and that's for 2 reasons. One, because we're testing 2 doses, and the other one, we do have a new drug, Relyvrio, so patients tend to be on all 3. We just want to make sure that we can see an effect on top of the effect that the other drugs will have. Is the CSF draw will be a requirement? As you heard in the video, being able to know if we are actually modulating this integrated stress response in the brain, the closest fluid to the brain is the spinal fluid, and it will help us understand if the drug is doing exactly what it's supposed to do there. So the spinal fluid will be very, very important for two reasons. One, to make sure that the drug is doing what it's supposed to do, and two, to make sure that this integrated stress response, is it stronger in certain people? Are the people with the highest stress response and the highest kind of decrease in that will respond to treatment? I mean, can we do what we call precision medicine and really target the treatment to the specific patient? And also the importance of this integrated stress response in ALS in general, just to get new understanding of this disease process. And
Well,
please,
thank you.
please, Merita.
No, I was going to just say, we want to thank people in advance for willingness to provide that. I think it's such an important fluid. We know that now from recent ALS science studies that they're finding new, really important disease biomarkers, but they're finding them in the spinal fluid, not in the blood. The blood is really just too far away from where the biology is happening. It is a safe procedure. It's one that neurologists do all the time, and anesthesiologists. And we're going to, I think this Thursday, I think Nick Maragakis from Hopkins is going to come and talk through more of the details of the spinal tap. And we have some people who have had spinal taps coming to talk to kind of explain it a little bit more. But it is really important for the study, and it is a requirement for the study because it's such an important part of figuring out the efficacy of this drug.
And I want to add something, that all the information we know about neurofilament, and I'm sure you all learned recently how important neurofilament are and how we can actually measure them now in the blood. All that information came from assessing neurofilament in the spinal fluid first. And we learned all that by knowing how it goes up, how it stays the same, how it kind of all the changes that the neurofilament does. And with now improvement in technology, we can measure very, very tiny amount of neurofilament in the blood. And we've been using the blood because we have the knowledge that we acquired already from the spinal fluid. So we no longer for neurofilament, for example, need necessarily to look at the spinal fluid, but for many, many other biomarkers, we're not there yet. We still have to do what we did for neurofilament to get there. Trying to see what else. Is the drug being studied for other diseases? Maybe Bill.
Yeah, maybe I can
Yeah.
help answer that one. No, we haven't opened any clinical. Well, we haven't studied this mechanism in other diseases yet, but we did recently open up a trial in vanishing white matter, and we did post that trial on ct.gov. So we will hopefully kick off that study soon.
And then I want to clarify, so the purpose of this drug is not to treat the symptom of ALS. The purpose of the trial, it's really to see if we can slow down the progression of the disease as we measure it with the ALS-FRS. So it's not just a merely symptomatic treatment. It's really trying to make sure we can slow down the disease progress. Let's see. Does the drug have a different mechanism of action than the other 3 drugs approved by the FDA?
Yeah, absolutely. And that's also what's exciting, is that in a way we will be testing it on top of standard of care, which is now, the Riluzole, Radicava, and Relyvrio. But it absolutely has a different mechanism of action. This is, to me, this is, as far as I know, there are a lot of trials going on. This is one of the first ones to be really targeting this TBP40, part of the TBP43 biology, and the first on this integrated stress response to go to phase 2 testing.
And Meredith, can I ask you this question? This is a very good question. They said if Relyvrio since Relyvrio is FDA approved, if you can't tolerate Relyvrio, can you take Cudkowicz instead over the counter?
So in this trial, and in fact, in many trials, Cudkowicz alone is over the counter is not allowed. And I know that sounds kind of odd because it's part of that Relyvrio, but that's because there's currently a clinical trial ongoing with Cudkowicz alone, and in all clinical trials, if there's something being tested in another clinical trial that you can't also take it because it makes it difficult to tell. It can make it difficult to tell if the new drug works or not.
And I want to clarify also that spinal tap will occur twice during the placebo-controlled phase of the disease, initially before taking the treatment and at the end of that period. That's the most important time. After that, it's optional. So if patient wants to do a spinal tap after that, we can collect it at week 28, week 52 at the end. But the most important one are the ones occurring during the placebo-controlled phase. And they will be done 6 months apart. I'm trying to read this one. So your mouse data showed improvement in function. Other drugs treatment in ALS simply shows slowing of decline. Are you expecting an actual functional improvement in ALS patient who already have a functional impairment?
Yeah, I think we kind of talked about this a little bit earlier. I think that we hope that this mechanism will slow down the progression and, you know, ideally stop the progression of ALS, but we don't know whether or not it would reverse or produce any functional improvements. I mean, one could theorize that there may be some cells that are sort of sick in the process towards dying, and this may potentially produce some minor functional benefit, but I think the main hypothesis is that would slow down the progression of the disease.
Very good. We addressed that TODCA by itself is prohibited. Only Relyvrio is allowed. And there's a question here, how soon after the site is activated that they will actually let you know you are in the study and can start? Also, one spinal fluid tap a month for 6 months. No, I'm sorry. I want to make sure only 2 spinal tap, one at the beginning, one at the end. I hope I made that clear. One spinal tap a month would be a little bit too much. And the other question is, you work with your site, and they should, when they're ready to enroll, they will let you know. Most sites usually have a list of patients who are waiting to get in, and they get given a call as soon as we are given the green light to go ahead. So just work with your site. They should be able to let you know right away.
I'll add that we do ask each of the sites to enroll 2 participants and then wait until they enroll the 3rd for us to just check that they're following all the procedures correctly. That's pretty typical in clinical trials. But once we've given them that okay, then they can enroll at any pace that they want and they have participants for. And we are really trying to support and encourage our sites to be enrolling you know, every day, if not, you know, every week, so that we can enroll in the trial you know, fast, because the faster we can enroll the trial, the faster we'll have, you know, the results of it. So I think this one will enroll quite well because we've already seen that activity in the first 18 sites that have opened.
And in case we haven't made that clear initially, the drug is administered orally once a day. It says, can I participate in the trial if I'm on a binder? I didn't see it well. And antifungal treatment. I think it's important to work with your investigator, I mean, with your site, and they will go over all your medication and make sure that there is no risk of taking the study drug with whatever you're on before. Remember, your safety is the most important thing for us. There is another question about sharing some anecdote from the phase 1. We understand it's ongoing and there is no actual data. This question actually came twice.
Yeah, I think it's I know it's very tempting to try to read into the tea leaves from an ongoing trial, but just you know, this is a small trial. It is ongoing. It hasn't been unblinded, so we really can't talk about any of the results that we might think we're seeing from that trial. Sorry about that.
Thank you.
You know, one person did write in their own anecdote about being their spinal tap in Regimen F. So I want to thank them for writing it, that they said it was painless and went well. That's good.
Yeah,
news.
it was, absolutely. Can you reconfirm that all 3 of the FDA drugs are able to be taken during the trial? Just wanted to confirm after the last comment. Yes, we want patient to be on a stable dose of these drugs for the Relyvrio, 30 days since starting. So it's going to create a little bit of delay. If somebody is just, for example, diagnosed, they need to start on Riluzole, Radicava, Relyvrio, depending on what the discussion with their neurologist goes. Not necessarily everybody needs to be on 3. I mean, that's really personal and different from patient to patient. And after being on these stable doses, that's when they could actually screen for the master protocol, and after that screen for Regimen F. If the patient decides not to be on any of the FDA-approved drug, we ask them to remain off at least for the duration, I mean, ideally for the duration of the whole study, but at least for the duration of the placebo-controlled phase of the study, so that we don't introduce any other bias. Can B12 shot be taken during the trial?
So we have not changed the criteria on that from the first ones, which is that there's a certain dose that people can take of B12 shots, which I think is less than 25 milligrams a week. But I'm looking at Sabrina to check if I got that number correct. Is that correct?
Yes, less than 25. Yeah.
So as a patient, will I notice a difference, or will we only know success from my lumbar tap? I think that's what it's meant. Well, usually when somebody is in a trial, it's difficult to see that there is a difference. First of all, you don't know that you're on placebo or on the active drug. And we usually try to wait for the end of the placebo-controlled phase and analyze the result to know if the drug has an effect or no. So in most cases, I mean, the answer would be no. The spinal tap would be helpful to see if there is a difference in response depending on patient. And usually that information will not be communicated back to the patient. So you will not know about the result of that either. And I think, and then go back and see if I missed any question.
I'll just add that, you know, the primary goal of the study is to tell the clinical effect of the drug. The biomarkers are really important and they're secondary, but they're to see again if there are perhaps subsets of people that respond differently. But it's the main goal is the clinical effects.
Yeah. I think I went over all the questions.
This was great. There were a lot of questions, really good.
I know. And interesting ones, really, very, very good questions. There was a question about a risk of infection when we do a spinal tap, and this is also need to be discussed with the investigator. There will be brochure to show how the spinal tap is done. And first of all, we want to make sure nobody will put any patient at risk. So if a patient has reason why not to have a spinal tap, we will not necessarily put them in danger. So if there is a reason, if their blood doesn't clot, or if there is any injury or something around the area, but the patient's skin will be cleaned very well and it will be done under sterile conditions. So the risk of infection from spinal tap is almost inexistent, especially the way we do it now. So I wouldn't have to worry about that. And we're not injecting anything in the spinal tap. We're just taking some of the fluid back. And that will be very well explained to the patient, to the participant.
this is for a different drug, but we've been following people in the tofersen, the gene therapy for SOD1 for more than 5 years who are getting infusions every month in the spinal fluid, and there have been no infections. It's really not a problem with spinal taps. The only reason sometimes we don't do spinal taps if someone is on an anticoagulation for, let's say, a heart disease or some illness where they can't come off the blood thinner, then we wouldn't do a spinal tap.
Let's see, there is more questions. Do people get better on the balance beams? I like this one. It's not one of our tests, but it's not a bad idea. Maybe we should think of that.
So thank you everyone for joining us for this hour and a real update, and any feedback on how the science is explained, please let us know. These were great questions. We will post this on our website. We'll see if we can work on the volume or the quality before we do that, but we're here for any questions. Please join us also on Thursdays, 5 to 5:30 Eastern Standard, and we'll be able to answer other questions about Regimen F and keep you updated on site activation.
Good.
Thank you, Bill and the team, and Senda for joining.
Thank you. Thank you all for the great questions.
Thank you.
Thank you.
Thank
Bye-bye.
you.